Our next speaker and the last in this session is Nico Borgsmüller, who works with Nico Borgsmüller at the ETH Zurich. And he's going to talk about how single-cell data somehow reveals deviations from clock-like evolution in cancer and also in healthy tissues.
Thanks, Magda. I try the next 50 minutes to walk you through this lengthy title, why single-cell DNA data reveals deviations from clock-like evolution in cancerous and also healthy tissue, a little bit more step by step. And I think I put myself into some good spot talking after David and Zenbai, because some of the stuff is already introduced. So, like David said, there's this debate on how tumors evolve over time, and this debate was already initiated in the 70s from the evolutionary dynamics community, when people debated how species evolve over time, and with the advent of single-cell sequence, or with the advent of NGS data, this debate made its way into the cancer community. I think the most prominent example is this 2016 paper, which you can see on the top left, where the group around Andrea Soteriva came up with some test which they applied to the TCGA dataset, and they claimed that they have identified neutral tumor evolution across different cancer types. And what they did was they looked at the variant allele frequency, and they claimed that in a certain range of this variant allele frequency, if you plot the inverse over the cumulative number of mutations, then you should get this linear relationship between the two. And what they claim is that if you do not see this linear relationship, then there's deviation from neutrality, so there's selection. And this sparked some quite controversial debate, because I think 2 years later there was another issue in Nature Genetics where 4 or 5 publications answered to the original one and objecting the findings. Once that some assumptions made do not hold, and second, that the statistic applied is not an appropriate way to do statistics. Like, you cannot prove selection by not rejecting the null hypothesis. And then this continued going on. The original author published a second method, which actually now works on this selection base. And last year, for example, there's another publication coming out which claimed that the original claims were actually true, and you could work with this 1/f frequency. So this is still an ongoing debate, which David said is not really settled yet. And all of this was done on biosequencing data. And like we heard before, there's some problems with biosequencing data. Within the individual subclones, we can infer from the variant allele frequency there's more going on inside these. But also, we cannot infer the evolutionary order of the subclones unambiguously. There's different ways which we can place the subclones in an evolutionary trajectory. Whereas with single-cell data, we have higher resolution, so we sequence the individual genotypes of individual cells, and this allows us to infer the branching order in the clonal evolution more precisely. But on the downside, the data is more noisy. We've got this high number of false negative rates and also missing values. And what we typically get at some point is this mutation matrix, where we have the mutations as rows and the columns are the cells, and then based on this, incorporating the error rates, we can infer the phylogenetic tree. And what we want to do now is to add to this whole neutral evolution debate a single-cell sequencing perspective, because everything so far was done on biotech data. And what we want to test is not really the selection versus neutrality, but we just want to test the null model, which is the molecular clock. And if there's nothing else happening, then the molecular clock is equivalent to neutral evolution. And the general idea is that mutations occur with a constant rate over time. So with every tick of the clock, mutations occur. Then these mutations do not change the underlying mutation rate, so they do not have any selective advantage. And if we then sample at one time point, this imposes some constraints on the phylogenetic tree. And that's what you can see here on the left. And the constraint is that the distance from the root to the leaves of the tree is equivalent for all of the leaves. which means, for example, there's the branch length of K1 here should be the same as the branch length of K2, K6, and K8 together. So that's our null model we want to test, and the alternative model. So like David said, there's like an indefinite number of selection models, so we just want to test the completely constraint-free model, where we assume each branch has its own underlying mutation rate, so it's no constraints at all. And here we can see, like just on one slide, what we are doing. So starting from a biopsy, let it be a cancerous or healthy tissue, we can sequence the data, apply some calling, and end up with this genotype matrix. Then we can apply some of the tools which are available, and also introduced earlier, and infer the phylogenetic tree and the error rates. Given the tree structure and the error rates, we can then map the mutations back to the tree. So we're observing, so the branch length is now the number of mutations occurring on this branch. And what we also can do is we can weight the branches. And the idea of the weighting is that different branches are more prone to errors than others. For example, you can imagine if there's true mutations on this branch are more prone to dropout events because they're just present in one cell. So if there's a dropout event happening here, this mutation is lost. Whereas the green mutation here, which is present in 4 cells, so 4 dropout events have to happen simultaneously to lose this mutation. So we can trust the number of mutations on this branch more than this one, for example. So,
sorry to butt in, but then you show that already 2 cells lost this mutation. I mean, there was a sequencing error, and the green star is not visible in 2 out of the 4 cells.
yes,
Yeah. Okay.
exactly. But yeah, to lose it, it should be present in, lost in all 4, whereas maybe there's a mutation which was just present in the red cell, and we do not see it at all because it's not sequenced. Yeah. So given we have the mutations mapped to the branch and the weights, then...
Nico,
Yeah.
so there's a difference between not seeing it and placing it on that edge.
Like if there's errors and we sequence this at least once, there's the probability that it's placed wrongly somewhere on the tree. But if it's on this branch and we don't sequence this, it's just lost.
Good job.
Yeah. So the number of mutations on this branch is like more error prone than the number closer to the root. Because for mutations to be lost here, more dropout events have to happen simultaneously.
Right, you have to lose it in both.
Yes, exactly. So given we have the weights and the number of mutations on the branches, then we can simply apply a Poisson likelihood where we weight the individual observations by this error proneness and find the maximum likelihood given the constraints imposed by the molecular clock or completely constraint-free. And this is a simple optimization problem, so we can obtain the maximum likelihood under these constraints. And then it's a nested model, so we can apply a likelihood ratio test to tell us how likely it is to see the null model given the data. And this is what we then did, first with simulation data. So we used this tool from David called CellQual, and it's simulating data based on a coalescent theory with some single-cell characteristics. So we sequenced 30 cells with a certain number of mutations, depth, and errors. And what you can see here now is the distribution of the p-values under the null hypothesis. So here we simulated clock-like data. And the rows is the increasing number of false negative rates and missing values thereby also. And on the first row, you can see 2 more or less naive tests. So one is a Poisson dispersion test, which just looks at the number of mutations in each cells, but it ignores the tree topology. And the second one is implemented in biotech-start, which is a classical phylogenetic tool. So it's designed for species data and assumes error-freeness, so that it's not able to deal with the errors which are inherent to single-cell data. And on the bottom line, you can see now, bottom row, our test. Once with a true tree, which we used for simulating and the true error rates, and once with the tree and the error rates inferred by the tool self-I. And what we can see, if we simulate data without any errors, then the classical phylogenetic test is already biased towards low p-values. So it's rejecting the null more often than it should. Whereas the Poisson dispersion test is more conservative. It's biased towards high p-values, which is in general not too bad. So it's not able to detect small deviations from the clock, but if we detect deviations, then we can be sure that they are true. But for our test, we can see that if we do not have any errors, then it is uniformly distributed, and that's what you would ideally want to see from an unbiased test. If we increase the error rates, then we can see also the Poisson dispersion test is biased towards low p-values. So what's happening, there's like a lot of deviations, a lot of difference in the number of cells, mutations in each cell. So these differences are misinterpreted as deviations from the clock, and this effect increases with the increasing false negative rate. Whereas our test with the weighting is also more conservative. So we're likely not able to catch up small deviations from the clock, but if we catch up deviations, then we can be sure that they are true. And if we increase the error rates, we can see if they are really high, then this effect also breaks. So with very high error rates, we have no chance to differentiate between single cell noise and true deviations from the clock. Then we modeled data under the alternative. So the idea is we simulate a tree topology, and then we pick a branch of the topology and multiply the number of mutations on this branch and the clade following this branch by some factor. And this simulates some sort of selective advantage. So the number of mutations has increased from a certain point in the topology. And we can see here once a multiplier of 2 and once a multiplier of 5. So that's like a rather minor effect, minor deviation from the clock, and that's like a stronger deviation from the clock. And in the columns, you can see how many cells are affected. Because it's not only the effect size, but also how many of the cells are actually affected by these effects. Because you can imagine if there's just one individual cell affected, then it's harder to detect instead of if it's like 50-50 split. Then it's relatively clear to detect this difference between the two clones. And what we can see is that if we have like just this small effect, then all in all, we're just able to pick it up in nearly up to 50%. But if at least 10% of the cells are affected, then it's going up to 75% and it's increasing up to 50%. And with a stronger effect size, we are able to pick it up in 70% if we just look at all simulations. But if we look at only the simulations where at least 10% of the cells are affected, it's 95%. So if the deviations from the clock are strong enough and they are affecting not just the small subsample, then we are able to catch them up reliably. And then we applied this method to all the single cell data, which we found freely available online. And yeah, one of the disadvantages, like we also heard, is that it's quite difficult to generate this data. So there's not difficult and expensive, so there's not too much available. And you can see most of this is whole exome sequencing. There's some whole genome sequencing data sets. There's different amplification methods. So MDA is the one which is most popular, but also Malbec. And most of these samples are from cancer patients, except this through neurons. And that's already now the results. So you can see that's all the cancer samples. And as expected, in most of these samples, we do reject clock-like evolution, with these three exceptions. For example, these two ones here are Malbec amplified, so it's not really ideal to call mutations. And yeah, this cancer polyp, we do not reject it like close to 0.05. So this is not really surprising because in most cancers we would expect some non-clock-like behavior. But most of these data sets also came with some normal cells. And when we applied the test to the normal cells, we could also see that we reject clock-like evolution in most of these normal samples as well. So in 6 out of 8. So in general, this means that also in normal tissue there's something going on which is not clock-like. And here you can just see now 3 examples. So that's from this one colorectal cancer patient. So in the normal sample, we can see the step-like behavior, which is likely caused by dropout events, like David explained earlier. And then down here we have clock-like behavior. So our test says this is clock-like because we downweight these branches because we do not trust them too much, but everything else going on looks clock-like. Whereas if we look at the polyps, there's some non-clock-like behavior going on in the down part of the branches. And if we go to the cancer samples, then we see that there's even a stronger effect here.
Just a quick question. Do you know if the normal is from blood or from the adjacent tissue?
I think it's, in this case, from the adjacent tissue. Yes, and with this, I briefly want to summarize. So we, for the first time, added a single-cell perspective to this neutral evolution debate on somatic evolution in cancer. On synthetic data, our method is superior to naive tests. It's unbiased in the absence of errors. But if there are errors, then it's more conservative. So if we find deviations from the clock, we can be certain that they are true deviations. And applied to biological data, we reject the clock in most of the cases of the cancerous samples as expected, but also in most of the healthy tissue samples. And maybe very briefly, we think that this is in accordance with the literature because there's different publications which also indicate that there's some selection or something non-clock-like going on in healthy tissue. And with this, I want to thank the collaborators working with me on the project. So there's Nico, who got COVID and is unfortunately not here. David, Monica is speaking on Wednesday, I think. And that's Jack Kupers from our group. Thanks.
Very nice job. I mean, using the p-values in the nested model. But as you mentioned in the last slide, molecular clock is not the same as neutral evolution. I mean, you can have even molecular clock. I will talk about this. You can have molecular clock assumption, and then you still can detect selection. I mean, because molecular clock is just the number of mutations per generation. But you can have more generation per time unit. And then in this case, you will still have the same number of mutations per generation, but you have more generation per unit of time. So you are growing faster, and that's why you're accumulating more mutations, even though the mutation rate is still the same. So
but this assumes like a change in the proliferative speed, doesn't it? Like that cells divide quicker, so it's some kind of selective advantage or some outside influence happening, right?
Yeah, but in this case, you cannot identify which one is the true. If you assume that the molecular clock is not constant, then you cannot assume. You cannot infer accurately that you are growing faster. If you assume that you're growing at the same pace, then you can have deviation from the molecular clock. So it's not the same thing. But you also mentioned in the last slide the Maxine Tarabishi contra article. They explained that about the null hypothesis. It's not equivalent. But
yes, But in the absence of anything else, clock-like equals neutrality. But it's hard to rule that out, right?
Have you defined neutrality? If you define neutrality that you are growing at the same pace, the same growth rate, then it's not because you can have more generation, you can divide faster, you can have more divisions in the same time unit, and then it is not you accumulate more mutation not because of the mutation rate is different you are accumulating mutation faster, but it's because you are dividing faster the cells.
Yes.
But it's
we don't know that if you are faster because that's what calculators want to know. But there are some cells dividing faster than others.
That would be some selective advantage, right? Or like some outside influence. Yeah.
Yeah, but still the mutation rate is the same.
Yeah.
stage-dependent signature, no? So you are accumulating mutation at the same pace, but then if you get a driver, then you grow faster. Yeah, this is what you are saying.
Yeah. No, he's saying that you cannot distinguish an increase in mutation rate from an increase in proliferation rate.
Yeah, exactly.
Well, I
But
mean,
you can-
a different mutation rate doesn't have to be functional.
But most cancers arise in epithelial cells, which by their nature are barriers, which means they are continually getting damage and continually being repaired. So when you have a damage event, you're going to get a lot of proliferation in that site. So it's more like bursts of changes. It's not like the cells are just happily in a constant environment. And also you've got like multiple tissue types like breast and prostate, where you have massive expansions during puberty, during pregnancy, during, so, it's a system that's continually changing. Over time, it won't always be the same.
And selection is something else.
Yeah.
No. We are totally aware of this, right, Nick?
Yeah. Yeah, it's.
About this. But the same critique applies to all the debate.
Yeah, of course.
Sure.
Because there is a problem of identifiability here. Of why? If a cell is dormant, for example, dormancy of cells, blah, blah. But the point here is that we can measure this, change in the accumulation rate, on the rate of mutations.
What's causing it? We don't know. But
Might be selection, might be other thing, might be like a environmental constraint or lack of resources for the cell to divide. But we just want to be able to call it, from there. And then interpretation is a different thing.
Can I ask something different? So, how much, in all this model, how much is impacting the number of cells that you are sequencing? Because obviously, the more cells you sequence, the more ancient is the last common ancestor, right? So your branching tree would be different, or not?
yes, I think the more cells, the better, obviously. I think especially with the data sets with small cells, you run into the problem that if you're unlucky, you get this just linear thing with individual cells branching out, so there's not much to tell from the small ones. So I guess, for the small, like 8, 7, 9, so there's little statistical power to tell anything.
So the age per se doesn't mean anything. What it means, because here it's about the relative age of different lineages.
Sure.
So the more cells, the better, because if your sample is, much larger, and you can catch things that you wouldn't otherwise. So you have 4 cells. It's very difficult to detect this unless you're really lucky to sample the 4 different lineages.
Yeah, but I was thinking more about healthy tissues, right? So with all the appearing knowledge about the clones competing in normal tissues, nothing to do with cancer. The more cells you sequence, the more you end up probably sequencing different clones.
Yeah.
Therefore, you are making maybe false assumption that they will derive from the same common ancestor. So you are rebuilding only one tree.
So
Why
there is always a common ancestor. There is always a common.
Yeah, I was about to say, there's always one common ancestor, right? So it's making sense to have one tree.
Come from bacteria, so or
The sample.
germlines.
So here there's
If you go back to Gamete, yes, there is only one.
Yeah.
there is a confusion here. One thing is the sample, a different thing is the population. So anything we can measure is the age of the deviations from the most recent common ancestor of our sample, whatever it is. The tumor might be or the.
Let Nico answer because it's his talk, it's not your talk.
Oh, yeah, sorry.
So he should answer.
You're right, sorry.
So,
I have a question for Nico.
in general, I think it's.
Just wanted to mention that we're out.
Sorry.
Yeah.
But if it's
In general, it should not matter because you cannot overestimate that there's always one common ancestor, and then you have different clones, and you sample these different clones at the same time. So if there's a molecular clock happening, then the distance should still be the same, independent of the number of clones you sample. I think that I rather see the problem with too few clones to sample, not too many.
So how advanced is your method for calculating the probability, the chance that you lose a mutation on an edge, right? That it moves somewhere else. And do you also, in that, consider the chance that a mutation is moved to this edge because of loss in the siblings?
Yes. So when we map the mutations, we do some soft mapping. So we based on the errors, we place them like where they're most likely, but with a probability. So this incorporates if they're placed somewhere else on the tree. So like the wrong placement is already incorporated when we assign the mutations to the branches. And for the weighting, we just look at each individual cell and calculate how likely it is that a mutation drops out from this branch. So we look at, for example, here we have to have one dropout event, and the other 4 cells have to be true negatives or missing values. And then we just calculate the probability that this happens on this edge, and we calculate the probability over all edges, and then we do some inverse variance weighting and get some probability based for the branches.
You don't take into account that the mutation can move?
Not when calculating this, but when assigning the mutations to the branches, we take into account that they can move. But not for the weights.
So the mutations are assigned sort of probabilistic?
Yeah.
With a certain...
All right.
I kind of questioned before as well. So because you have a very small number of cells, I just wonder what would you actually... Did you do some simulations to see how that affects your results in a way?
Yes. I actually ran this like 2 weeks ago. So we simulated 100 cells and we subsampled 10, 20, up to 90. Yeah, but I just had a brief look at it.
Okay.
So it's, yeah.
And maybe you asked the same as me.
Yeah. All right. Thanks.